I was getting some similar questions from different people, so rather than respond to the same thing over and over individually, I decided to put these responses into a video. So first off, how do we find the expected values in a question like number 29? I have the question copied and pasted here. I'm not going to read the whole thing, but some key items are that we have 3 groups of women, 50 women in each group. They each receive a treatment. It's either cranberry juice, lactobacillus drink, however you say that, or a control group that drinks neither of those 2 beverages. So to start off, we can set up a table, and that's what we would want to do first for a problem like this. We're tracking to see whether or not they develop at least one infection. Okay, so we have infection, no infection on one side of the table, and our 3 treatments across the other side, 50 women in each group. After that, we can fill in this information that it gives us. So in the cranberry juice group, 8 people developed an infection, 20 for the lactobacillus, and 18 for the control. Okay, we can figure out these other blank boxes at this point, total up the whole row in these columns over here, and we see that regardless of which way we look at it, there's 150 people in the table. That's the way we verify we did it correctly. At this point, we have this table, and we want to find the expected value for each of these cells. Short answer, it's the row total times the column total divided by the grand total. So if we wanted to find the expected value for this cell right here, we take the total number of instances in that entire row going across, we multiply it by the total number of instances in that entire column, so 46 times 50, and then divide by the grand total, 150. That's going to give us 15.33. That's actually going to be the same for all of these rows across the top, because no matter which one we look at, we have row total of 46, column total of 50, divided by 150. That's not the case in every table, but it is the case in this table because all of these sample sizes are the same. Longer answer, let's look at just this information on the right here. Okay, 46 out of the 150 people developed an infection. That's across the entire experiment. That equates to 30.7%. So if the infection rates and non-infection rates are the same across the groups, that's what we're testing, we would expect 30.7% of each of these groups to develop some kind of infection. 30.7% of 50 is 15.35. There's a difference because we rounded this, but otherwise we would get the same thing. If these sample sizes were different, okay, we would just take that 30.7% that we got from this number out of 150 and multiply it by whatever our sample sizes are. That's going to give us expected values for these cells. Note that when we're doing this, 30.7% is just this fraction. So we're really doing that fraction times the sample size, which is row total times column total over the grand total. Next question, what's actually happening when we calculate a chi-square test statistic? So at this point, we figure out the expected cell for this first cell here. We got 15.33, and we also noted that this is going to be the same for these 2 cells as well, because for all of them, row total times column total over 150 is the same thing. All right, so now what we want to do is we want to find a chi-square value for each of these cells. And that's given by this equation right here. Observed minus expected squared divided by expected. So if we did it for this first cell, okay, it's 8 minus that 15.33 expected value. Square that, divide by expected. That's this one. If we do it for this middle one here, okay, we get that equation. This one at the end gives this equation over here. Okay, these are all really similar because these expected values are the same. That's not always the case for every chi-square test. Simplify those and we get chi-square values for each of these individual cells. The chi-square value for the entire test is going to be all of these values added up along with the values in these cells, which I'm not going to walk through. All right, but we can make note of the fact that this 3.5 is a lot bigger than these other ones, 1.52 and the 0.47. And the reason is that this 8 is farther away from the expected value. Okay, so that makes that chi-square value larger. It's going to make the residual larger as well. If we were to look at the residuals, there's less people than we would expect in this cell, would have a fairly large residual because that's farther from the expected value than either of these two cells, which both have a little more than expected. If we did all of these cells and we added them all up, we would get a chi-square value of 7.776. We could use the chi-square CDF, right, same place you would find normal CDF and TCDF, to actually get a p-value from this. This is always the lower, and then upper we would put in a big number like 99 or 999. How do I put these homogeneity and independence tests in my calculator? This is harder because I don't have a simulator on my home computer to walk you through this. I also don't have one for the 89 either way. So I have videos from YouTube that walks you through each of these. First one is for TI-84. Second one's for TI-89. If you watch these videos all the way to the end, you'll see how to view the expected counts in the matrix that's created when you do the test. Last question, how do we tell what type of test it is? I'm going to walk you through a whole bunch of different examples right now because there's a lot of different things that we look for. In this first example, this is probably the easiest one to identify because we have just one set of data here. We have one sample, it's in a single row. The only thing this can be is a chi-square goodness of fit test. Okay? For us to do either homogeneity or independence, we need an additional row. For this problem, we would check to see, do the number of red results match up with what they're supposed to be given 18 out of 38 are red. So that would give us a percentage. Is 85 out of whatever this total is, 200. Okay, close enough to this along with these two when we look at these, where our p-value is not too small. This one's about the first million digits of pi. Again, we really only have one data set here. Okay, this row is not data, it's just labels for that data. We have 99,959 zeros in the first million digits of pi. The question is asking, are these numbers uniformly distributed? We can see they're all around 100,000. Okay, is it within an expected degree of variation, or are some numbers occurring more often than other ones to a statistically significant margin? Back to the question at hand, we have one set of data. This is a chi-square goodness of fit test again. Next up, this is talking about seagulls landing on a couple different set of options. We have sand, mud, and rocks. We're counting where they land. The actual data is down here. Okay, again, we have just one set of data, and we're comparing it to a set of percentages here. Okay, our data is a single sample. It would be in one row. Rocks, mud, sand. Okay, does it match up with these percentages? Again, chi-square goodness of fit. When we look at this one, we see right away this is a table with two dimensions, male versus female on the left, three different options across the top. This can't be a chi-square goodness of fit test because we don't have just one set of data, one row or one column. So this is either independence or homogeneity. Okay, there's a giveaway in the actual wording. Anytime we see this word right here, that's a test of independence. If it's asking about is there an association, it's just another way of phrasing, are these variables independent of one another? The one that we did at the start of this video said that we have 3 different groups of women, 50 women in each. We want to see, are these different groups the same in terms of developing infections? This is a test of homogeneity. Anytime we have more than one sample and we're comparing those samples, that's a test of homogeneity. Again, we have a big table so we can rule out goodness of fit. We can't really tell if the survey is made up of a number of different samples or a single sample. We're going to check the wording. It's asking us to investigate the differences in different age groups. When we're talking about things being the same or different, it's a test of homogeneity. Here's another one. Is there evidence that the distribution of outcomes is not the same? Okay, are they the same? Are they different? Test of homogeneity. Is there evidence of a difference in the proportion of crimes, the type of crimes committed during a full moon versus not a full moon? Again, talking about differences, homogeneity. Lastly, in this one, the wording doesn't give us any hints. What does give it away is that we only have one sample. This table is created based on a sample of 1,178 births. We're not seeing is one sample the same as the other one. Instead, we're taking the sample and we're measuring two categorical variables. We want to see, are these variables affecting one another? Is there an association between getting an epidural and the effect on a child breastfeeding 6 months after birth? This is a test of independence.